So hi, and welcome to the first pre-recorded video on viruses. I'm going to have two parts to this. The first part is on viruses. We've already talked a little bit about viruses, how small they are, a little bit about human herpes viruses. I want to talk a bit more today about life cycles and their structure and what they do. Part two is going to be on SARS coronaviruses and COVID-19. So let's talk about viruses and how they are built. First thing to know about viruses is that they have a genome, and the genome can be DNA or RNA. It can be single-stranded or double-stranded. It turns out there's actually quite a variety of genomes that viruses can have, and we'll talk about that a little bit later on. The other thing that all viruses have is a capsid. Capsid is basically a protective layer. It's a protein coat made of a bunch of different proteins, and this is it. All viruses have this for the structure. Some of them, this is all they have. Quite a few viruses actually do have additional features, and I'll mention a couple of them right now. One additional feature is something called an envelope. This is actually basically a membrane that the virus takes when it buds or escapes from the cell that it's been infecting. It's not found in all cells, but pretty common in animal viruses. And part of that envelope is sometimes something that we call viral glycoproteins. And so these are glycoproteins. Remember, glyco means carbohydrate and protein means protein, so it's a combination of both. Sometimes we call these spike proteins. Some of them kind of look spiky. And this, of course, has come up a lot when talking about SARS coronaviruses because they have spike proteins. Here are a few different viruses. You can see they all have some of these features. They all have genomes, so DNA or RNA. They all have capsids. Capsids are protein coats. And the last one on the right also has a membrane. And so I'll show you a few different virus structures here because they do come in many shapes and sizes. So one type of viruses are these helical viruses. Just as they sound, the genome is kind of wrapped up in a long helical strain. You can see the tobacco mosaic virus and the Ebola virus are an example of that, and it still has a capsid, which is kind of in a tube-like or a strand-like structure. Ebola virus, in fact, is long enough it's kind of spaghetti-like and sometimes ties knots on itself, which is kind of really fascinating. Many viruses are polyhedral viruses. The capsids form these little triangles, and little triangles basically form a three-dimensional structure with a hollow part in the middle contains the genome. So if you have a small genome, you need less triangles. If you have a large genome, you would need more triangles in order to do this. This particular shape is something called an icosahedron. And you can see there's two examples, the poliovirus and adenoviruses are an example of that. Adenoviruses are a variety of viruses. Some of them cause mild things like colds and some respiratory infections, usually pretty mild infections from adenoviruses. Some viruses are enveloped, as I mentioned. Actually, quite a few animal viruses are enveloped, and the envelope is basically a membrane. Not quite like a membrane that we have because it's not performing all the same functions, but it is an extra coat on the virus. You can see I have three examples here. I have HIV and influenza viruses. These viruses I usually spend a lot more time talking about in Biology 107, but the coronaviruses, of course, taken a front seat since then, and I don't really talk about HIV and influenza the last year and a half anyway. So we're going to be talking a lot about SARS coronavirus 2 in part 2 of this pre-recorded lecture. There's another category of viruses that sometimes we just call complex viruses. It means they have kind of extra bells and whistles and extra features. So if you take a look, this is a bacteriophage. So bacteriophage, by the way, bacterio means bacteria, and phage means eating. So what we have here is a virus that eats or infects bacteria. So it infects E. coli, for example. You can see this has a head, and the head structure is basically a polyhedral kind of virus. It has the genome, and the genome is surrounded by that capsid, that protein coat. But it also has this big tail structure. And the tail structure is kind of like a giant syringe. And what it is going to do is inject the DNA from this virus into the host cell. So we'll come back to bacteriophage in a moment. Or right now, forgot I found this little animation on the internet. Yes, this is what the internet is for, to have all the silly and cute things out there. I thought you might like it. So as I mentioned, they come in all shapes and sizes. You can see there's E. coli in the center of the slide, and the bacteriophage in comparison, not much bigger than the hemoglobin protein, and also not much bigger the flaviviruses, which cause things like yellow fever and Zika. Some of the larger viruses, like smallpox, over here is not as big as E. coli, but getting there. It's a very large virus, has a very large genome as well. So I want to talk a little bit about how viruses do what they do. One thing that's worth mentioning is that all viruses have what we call as a host range. So what kind of host it will infect. So host can mean two things. It can mean the type of organism and the species. It can also mean the type of tissue. So you can see I have an example there is HIV. HIV stands for human immunodeficiency virus. You may or may not know that. Human immunodeficiency virus. And it infects exclusively a type of white blood cell called the CD4 T lymphocyte. So these are cells are important in your immune system. So if you get HIV infection, it's going to kill immune cells. And if the disease gets to a certain stage, it turns into something that we call AIDS, which is autoimmune deficiency syndrome. And it can be a very bad thing to have because your immune system's not working properly. So very, very specific both to human, the organism, and also the cell type. We also mentioned T4 bacteriophage. We mentioned bacteriophage will infect things like E. coli. The T4 is specific to the organism E. coli and won't infect like salmonella or something like that. Some viruses have a bit more of a broad host range. So influenza A virus will infect humans, pigs, and birds. So multiple species there. Some are extremely promiscuous. So the rabies virus, for example, will infect you. If you are a mammal, it can probably infect you. And so very, very promiscuous. So I want to talk a little bit about bacteriophage because you're going to see them in the next few pictures. Bacteriophage are very, very well studied. As you can imagine, since they're only killing E. coli and they're not causing any human disease, you don't have to have all sorts of biosafety hazard levels and all those kind of things. And so much of what we've learned about viruses, we learned first in bacteriophage. There's an image there of T4 bacteriophage infecting E. coli, and E. coli is not looking too happy, is it? There are many types of bacteriophage. I want to share a few pictures with you. There's the lambdaphage, PM2, MVL2, and my personal favorite is the corn dog bacteriophage. Very, very cute. So what I want to do is show you what viral life cycles look like. And if you take a look here, this is showing a bacteriophage infecting E. coli or some other bacteria. So the first cycle I want to talk about is the lytic cycle. So the lytic, that word lytic is very similar to the word lysis, and it means something like breaking. So you're going to see at the end of this life cycle, it's going to break out of the host cell. The very first step is the attachment. So something in these tail fibers that is going to recognize something on the surface of the host cell. So on the, it's probably some sort of glycoprotein on the surface of E. coli. And this is why viruses are very specific to which hosts they are going to be binding to. The first step is attachment. This is followed by the virus basically getting its genome inside of the host cell. In the case of T4 bacteriophage, it's actually a very hostile takeover. It actually destroys the host DNA at the same time. Very, very nasty. But you can see there we have the genome, in this case DNA, is getting into the host cell. So DNA, of course, is instructions. And those instructions basically say, okay, you're going to make more viruses from me. So that DNA codes for things like the viral glycoproteins or the capsid proteins, and it's going to make new virus parts. just using the host machinery. So the host enzymes, RNA and DNA polymerases, and ribosomes, and so on. Basically, after that, it starts assembling, and it's going to produce fully functional viruses. And then it's going to get to that lysis part, where it breaks open the host cell. And if this is a bacteriophage, it takes about 100 minutes, and you're going to release 100 to 200 more virus particles. So you can see that what's happening here is the virus is killing the cell. And this is actually how a lot of viruses cause disease, is they destroy tissues. So sometimes it might cause a blister, like a cold sore, and it's kind of annoying. If you've had a respiratory virus, it might be infecting cells down your respiratory passages. That might lead to a cough or chest pains and things like that. And then some, of course, do much more damage, can actually kill you, unfortunately. But not all viruses do this. So I want to talk about the lysogenic cycle in a moment. I do have a little video here for you, though. So here's a little video of a bacteriophage infection. And so if you take a look at this video here, you can see there's a bunch of E. coli, and they're swimming around. And we also have these little viruses are swimming around. And there's one there. It's going to recognize something on the surface of the bacteria, and it is going to bind and attach. Take a look. There's the DNA. The DNA is green. It's going to get injected into the host. Here goes. So we're going to skip ahead 100 minutes to the point where the new viruses are escaping from the host cell, and there they go, and they're going to go infect more bacterial cells. So that's the lytic cycle. Now I want to talk about the lysogenic cycle. So lysogenic, I'm not sure exactly what it means. It means something like sleeping. So in this case here, the very first step is the same. You have a virus particle, it's injecting its genome into a host. Now at this stage, it's not going to destroy the host cell necessarily. And in this particular virus, it goes into something called the lysogenic cycle. So we'll come back to what the lytic cycle there on the left in a moment. So this lysogenic cycle, if you take a look, we've got the viral genome there. That's that little blue thing. And you've got the host genome there. And what they do is they integrate together. And so now the virus genome is part of the host genome. And if this is a bacteriophage, we call this a prophase. If it's another kind of virus, we call it a provirus. So if that was in an animal cell, we would call it provirus. It's a good word to know. Could end up on your midterm. And then what happens is the cell just lives. It divides and undergoes binary fission or mitosis, and the cell just replicates. So this is one way that viruses hide. This is kind of similar to what happens with HIV. People who have HIV have a lifelong infection because it can hide in their immune cells. And it's kind of impossible, at least with current technologies, to get rid of the virus when that kind of thing happens. Eventually, though, what often happens is the virus does start to express itself, and that's when we have the lytic cycle being induced. In the case of cold sores, it could be a damage or weakened immune system, or in some cases, ultraviolet light that actually stimulates the virus to come out of hiding. And there it goes. And so then it goes through a lytic cycle. So in this case here, it's doing what we did before in the lytic cycle. It's expressing the virus parts, so proteins, making new virus genomes, eventually breaks out and starts the cycle again. So lytic cycle, lysogenic cycle, kind of basically just different strategies on how to do what you do as a virus. Sometimes you wait a little bit and you sleep and you go dormant. Sometimes you break open the cell right away. Most virus life cycles follow, one or the other of these or some sort of modification of. So that's why they're worth mentioning. So one other thing worth mentioning is that these life cycles I just showed you were shown in bacteria. When we get to animal cells, there are some differences. Animal cells do not have a cell wall. Bacteria do. That actually makes it harder for the viruses to get into a bacterial cell. But animal cells do have a nucleus. And so some viruses have to get into the nucleus. Not all of them. Some replicate in the cytoplasm, some replicate in the nucleus. It kind of depends on the actual virus and what they need to do. And much variety on this kind of thing. And we'll take a closer look at what's going on with the SARS coronavirus in the next part of this lecture here. I have a little video I'll show you of an animal virus life cycle, and there's another narrator, so I'll let him speak and tell you what's going on.
Here's the virus particle with its envelope and proteins and genome. And you're going to see this represented as being within a pipette that's going to be dropped onto a petri dish containing human cells that are going to be susceptible to infection. Here's a blow-up of a single cell. Keep your eye here. These are the cell surface receptors. Here comes the virus particle that's going to bind. Here comes the disassembly stage. The genomes will, in this case, enter the nucleus. Not all viruses have to do that. And now you see replication. Here comes the assembly reaction. And these cells are now, the capsids are going to now go to the cell surface and bud. So what we've just witnessed is a single cycle of viral infection. A single particle infects a single cell and gives rise to many progeny. On average, 100 or so progeny per cell. Some viruses up to 1,000 or even more.
Okay, so there were the kind of some of the basics around virus life cycles. Here's an image from your textbook that's showing what's going on with a kind of an animal virus life cycle. You can see it's all the same stuff. Basically, it's getting the genome in. There's different ways to do it. It can get in by endocytosis or other methods. The viral genome is copied and read by the host enzymes and makes more virus proteins. You can see there's the capsid proteins being made there. There's the viral genome being made there. Some of the viral proteins are made in endoplasmic reticulum. And so depending on the virus, there's a variety of ways to do this. And in the end, you get your new virus over here. This particular one here seems to be budding off, and that seems to be its method for getting out of the cell. So like I said, we'll take a little look at coronavirus life cycle in the next part of this lecture and get a few more details out of that. At the moment, it's just good enough to know they need to get their genome in, and the genome carries instructions for making new viruses. So I want to talk a minute or two about viral classification and these viral life cycle strategies. So you may remember this particular slide I showed you way back, I think in topic 1, about how cells take DNA, and from the DNA they make RNA, that's called transcription. And then from the RNA they make proteins, that's called translation. Well, it turns out that viruses, well, they don't have cells, they're acellular. And so they do things a little differently. In fact, viruses have 7 different strategies for doing this kind of thing, and they don't have to do it the same way that cells do it. I'm going to just show you very quickly the Baltimore system of classifying viruses. It breaks them into 7 groups based on their genomes and how they do and replicate themselves. First group here is group 1. They're double-stranded DNA viruses, and they use the DNA to make RNA, which is used to make proteins. Group 2 are single-stranded DNA viruses, and they have to make their DNA double-stranded before they can go through the whole system. Group 3 are double-stranded RNA viruses, so they're skipping the DNA step entirely. Groups 4 and 5 are single-stranded RNA viruses. This includes the coronaviruses, which we are going to talk about, and the influenza viruses, and there's plus and minus stranded viruses. I'm not going to get into the particulars there. There's also HIV. HIV kind of does something weird where it starts off with RNA, and then it uses RNA to make single-stranded DNA, and then that's used to make messenger RNA. Kind of weird, but it's working for HIV, so we know it is a valid strategy. And then lastly, there are some other double-stranded DNA viruses that kind of have an extra step in it, so they're their own group 7. So what's the key point here? The key point is that all viruses have to make messenger RNA, and there's different strategies on doing so. You can have DNA or RNA genomes, and those genomes can be single-stranded or double-stranded. So virus classification in reality is a bit of a mess. You can classify them based on their genome. You can classify based on their structure. Are they enveloped? Are they not? Sometimes they're classified on their host range, meaning insect viruses, plant viruses, and so on. If you care, if you want to make a difference, you could always join this committee, the International Committee on the Taxonomy of Viruses, and maybe make a difference in terms of how these things are classified. So here's some examples of some animal viruses, and from all the groups, you can see double-stranded DNA. We talked herpes viruses already. Right here, this includes, as I mentioned before, cold sores, genital herpes, chickenpox, and so on. We can have single-stranded DNA viruses, double-stranded RNA viruses, single-stranded RNA viruses, and this includes the coronaviruses. So much more on these in the next part of this lecture. So lots of hints of things to come. There's rabies viruses, retroviruses. So human viruses come from all the categories, which is kind of the point I wanted to make. So this is the last slide for this part of the lecture. Kind of just a little matching question here. See if we can figure out what is going on here. You can see I have the macromolecules on the left, and I have the some virus parts on the right. So let's take a look at this. First of all, what of these things is proteins? There's two answers to that. I didn't mean to have two answers, but the main answer is enzymes. Some viruses, I didn't really talk about that this semester, but some viruses have their own enzymes that they're bringing to the infection, like HIV is a good example of that. And enzymes are made out of proteins. Also, you can see there's the protein part of glycoproteins. Those are also made out of proteins. Nucleic acids, in this case, we're talking about the genome. And remember, the genome can be DNA or RNA, and it could be single-stranded or double-stranded. Lipids, we are talking about the envelope. So envelopes are membranes, and they are, of course, made out of phospholipids. And carbohydrates, we are talking about the glyco part here. Remember, glyco means carbohydrate. So which components are part of all viruses? All viruses have a genome, RNA or DNA, and all viruses have a capsid. Capsid is a protein code. So those are made out of proteins. All other things are not found in all viruses, just some. So that's the end of part one. Stay tuned for part two.